Welcome everybody to Anatomy and Physiology 101. This is the first lecture for the first chapter, for our semester. We will be working with Erin Ammerman's Human Anatomy and Physiology textbook. We're following along with her organization of that textbook. I'm going to record through this Zoom session an outline lecture of the first chapter. And then there'll be Zoom recordings for each chapter to go over the material from your lecture textbook by Erin Ammerman. To participate in this lecture, you need either a notebook paper so you can write down some ideas, or just listen the first time through and then maybe go back and write down your notes. But I also highly recommend that you take a look at my chapter outline that I've made for you. And you will find that in Ivy Learn. So that's what you should be seeing on your screen right now is I'm in the Ivy Learn course in modules. If you scroll down and find the chapter 1 module for lecture resources, you'll find a document right here, .doc. This is a Word document that has lecture notes for chapter 1, an outline that I made. You'll also find a link to the PowerPoint. You're going to see the PowerPoint in the video today. I'm going to share it and show it on the screen. So if you want to come back and just look at the PowerPoint yourself without the video, you have that document or that file here. You have access to that file. This is a PowerPoint that I made, customized for chapter 1. The publisher also makes PowerPoints. They have a lot more slides in them. They're a lot more detailed. So if you are a detail-oriented person and you like to get everything from the chapter, to some people it just makes more sense. They understand the concepts better. That is absolutely a good thing to look at. So for this first couple chapters, I think I put the publisher's PowerPoint. If you need help finding those for future chapters, let me know. The publisher also makes a lecture outline, and you might find that very useful while you're reading. Some students like to have that hands-on work while you're reading the book. You just need something to wake you up every couple, keep your brain engaged, keep you going as you're reading. You might want to print out this lecture outline. It's again, a Word document, and it goes through the entire chapter and has little blanks for you to fill in and will help you as you're reading the chapter. So I have those on there for the first couple chapters. If you want more, they are available through Mastering A&P. If you have trouble finding them, let me know. For each chapter, though, I have made a summary outline in the notes that goes along with the PowerPoint that I made. And I'm recording this on Zoom. Obviously, you're watching a recording, and I post all these lecture recordings for the spring 2021 semester. They're posted in a Google Doc, so you can see this Google Doc gives you the links to the recordings on a list. So that recording, once it's processed, will be posted here. So I've already opened this document. Again, you'll want to come back to this, the lecture notes document. I've already opened it in Word, so it should look like this. And you might work with this on your computer screen and be able to type in it, or you might want to print it out and hand write your notes in here. Or like I said, if you prefer to take notes in a notebook, on notebook paper, that is perfectly good as well. That is a great strategy. But if you'd like, you can write directly on these pages. If you prefer to take your notes differently, that is fine. I do recommend, though, that you read through these notes. Look at them at some point just to give you an overview, and make sure you have, hit all the highlights that are in here, because this is what I look at when I make test questions. So I come back to here and say, okay, was it in the notes? If it's in the notes, I talked about it in the lecture recording, this is what I'm going to test on. So you'll want to come back and just kind of double check this and look it over as you're studying for the test. Okay, so that's our notes packet. And in a minute here, I'm going to switch over to the PowerPoint, and you can be taking notes while I'm going through the PowerPoint slides. I am not going to go over, however, the first module. So chapter 1, it's broken down into different sections or modules. Module 1.1 is titled How to Succeed in Your Anatomy and Physiology course. I'm not going to lecture on that, but you should read that absolutely and learn about special strategies for studying and learning anatomy and physiology. It's very different than studies that you've done for a math class. This is a different way of studying than math class. This is different than an English class or a psychology class. there are different strategies that you might use. Some of them are same, but some strategies might be very different. So this might give you some new study strategies to try, some ideas that you might want to try. And this is so critical because this course is foundational. Probably, if you're looking at health sciences career, this course is probably a foundation course for you, meaning we're going to begin to learn the basics here, and then so many other classes that you take will build upon that, like layer after layer after layer, and they'll be presented similarly. So some of the strategies that you learn here are the strategies you're going to carry forward and use for success in so many other classes. So again, like, and in fact, I often tell students, like, this is so important that you read this first section and you work on this semester. In fact, I often tell students that maybe it's as important, sometimes I think more important, that you learn how to study in Anatomy 101 than you even learn all the stuff that I'm going to tell you. Okay? Because if you learn how to study, then even if you don't get it this semester, you're going to learn it in the future. You'll figure out how you're going to learn these things. So two very important goals this semester: getting some foundational ideas, but also learning how you're going to acquire more information and more knowledge through your own study strategies. And they're different for different people. So they're going to explore some different possibilities in this first module, and you are experimenting and figuring out what the right study strategies are for you. Okay, so again, I'm not going to go over that module. I'm going to start with module 1.2 in my PowerPoint and talk about anatomy and physiology, introducing these topics. So I switched over to the PowerPoint slides. Okay, we're going to start here. Okay, so chapter 1, this chapter, just a heads up, is very broad. It's an introduction to anatomy and physiology. There's not like one organ system. We're not studying blood, we're not studying the reproductive system or the bones. We're studying a little bit of everything. They're kind of showing you a little bit of everything in this chapter. So it's a little overwhelming. Sometimes students are like, oh my gosh, this class is so much. Well, we're showing you a little bit of everything in this first chapter just to introduce it. We're not expecting you to know every detail about every organ. They present all the organ systems in summary in this chapter. I'm not expecting you to know all those details yet, but to get at least a first look at them so that they're familiar. We will go in more depth on each organ system beginning a little bit later in the semester. And we will continue that into Anatomy 102. Anatomy 102 helps us have enough time to cover all the organ systems because we can't do it all in one semester. Additionally, this chapter will give you a framework. It kind of show you like how we're going to approach things, and some foundational concepts that, just overlie, like you have to understand, kind of basic layout, structure, and organization of the body so that we can add more details to that. So introduction. All right, so first of all, I want to point out that what we're studying in this course, let me get my pointer here, my laser pointer, anatomy and physiology is what we're studying. Two different things, actually. We're studying two different topics, but they are both subdisciplines of biology. And biology is the study of living organisms. So if I'm a biologist, I don't just study anatomy and physiology. I don't just study humans. In my educational years of study, I was very interested in plants and ecosystems and microbiology, bacteria, all different types of living things that I studied in my biology program. Anatomy and physiology are just specialized parts of biology. We're not going to look at necessarily ecosystems or populations, groups of people. We're not going to study groups of people. We're going to study and there's anatomy and physiology of like animals, like you might say anatomy and physiology of dogs and cats if you want to be a veterinarian. Most of you are probably enrolled in this course because you're looking for a human health science career. So you're going to focus on human anatomy and physiology, looking specifically at the structure and function of humans. So just a subdiscipline of biology, studying living things, in this case, humans. But first of all, I just want to point out the difference between life science, studying living things, versus natural sciences like geology, chemistry. Chemistry can be life or non-life, but still natural physics, for example, is natural sciences, but not necessarily looking at living things. So how would we know? It's kind of important to, recognize, okay, all the things at the top here, this baby, this dog, the bird, the bug, the mushroom, the grass behind it, these are living things, versus, like in physics, you might study heat, okay, or a volcano, okay, in earth science, wind, so forth. These are non-living things, wind and water and flames and rocks and stuff. Certainly part of our world, but not living. What's the difference? How would you know the difference? And this might seem like extremely obvious, like, okay, how basic are we starting here? Like, what's alive? But, I'm sure you know the difference, but how do you know the difference? Can you put words to it? Can you define what life is? That's pretty important. And this is really, really important in health sciences, because the goal of health science is to maintain life and good health, like healthful living. Okay, this is the difference between, your patient is alive or your patient is dead. I mean, how, it's just getting real here with you that, you've got to know, like, how are you going to say, how does a doctor pronounce a patient dead? Like, what are those characteristics? What are we looking for? How do we know if something is alive or not? Okay, take a little time to think about that here in this beginning chapter as we're starting out. What are we going to focus on? What are those characteristics, okay, that we're going to focus on? How do we identify them? How do we measure them? How do we understand how they happen? How do we maintain them? Because in healthcare, we're trying to maintain that living state, keep people alive and healthy. What are we working toward? What should we be examining in our patients? So, characteristics of living things. Some of the things that, you might say, well, I know that's alive because it moves around and does stuff, right? Okay, so movement. Yeah, that's a characteristic. If your patient is unable to move and, are they alive or not? So movement is a characteristic that we typically associate with living things. Now you might say, well, no, wait, the plant was a living thing, the mushroom was a living thing, they don't move around. True, but movement, and I'm going to type some things here and you might want to add some notes here. Movement can be movement of the organism or within the organism. So, for example, a tree doesn't get up and move around, but the sap, within the tree moves sometimes from, the branches down to the roots, or from the roots up to the branches. We see, that type of movement. Water moves from the roots up to the branches and the leaves and so forth. So there are movements within. Even a tiny bacteria. A bacteria is made up of just one cell. Inside that cell, there are molecules moving around. Sometimes there are whole organelles or structures within that cell that move from place to place. Sometimes bacteria can even move itself from one place to another. So you may not always see the movement, but all living things have some type of, either external motions or movements within the organism that they're capable of. Irritability is another characteristic, and yeah, I can relate to irritability, right? Sometimes on a Monday morning, I get this characteristic, right? But actually we have this characteristic all the time. It's really just another way of saying, or also known as responsiveness. Responsiveness or ability to respond to what we call stimuli or stimulus. Okay, I mean, if you think about it, when you're irritable, that usually means like somebody says something and you react like strongly, right? So it's about that reaction or that response. And this doesn't always have to be negative, right? It can be a positive response, like, you know, you want to give somebody a hug, you're so happy, right? Because they did something nice. And so any type of response. Now, and these can be responses not just to like the way people act, but how about to the changes in your environment, like getting very cold out, okay, and you might shiver to try to warm yourself up, or it's very hot out, you might start sweating to cool yourself down. So, you know, running out of nutrients, you get hungry, you eat. So it's responding to stimuli, changes outside the body, changes inside the body. It's that ability to respond to different changes that happen to us and around us that affect our function. And all living things, even that bacteria, okay? Some bacteria, when there's not enough water around, they encapsulate themselves. They go into like hibernation, just like like a bear would hibernate in the winter. Bacteria can like hibernate. They put a capsule around themselves and they can survive. They go in this really low active state. And even though there's no water, they've like put a sealant around themselves to hold in any moisture that they have, and they can survive, you know, for many, many years like that. So they're responding, right, to changes in their environment. All living things have that ability to respond. All living things, from bacteria to trees and mushrooms and blue whales and humans, we all have an ability to grow and to develop. And these are actually two different things. Let me put this down here. Growth is an increase in size. You know, baby blue whales start out small, right? And they grow up into big mommy and daddy blue whales. So they'll get bigger during their lifetime. And even bacteria, when a new bacteria is formed, it is often slightly smaller than it's going to be later on. So they have some ability to increase in size. Development is a change in structure or form. You could say the form of something. And you undergo development or changes like look like what you were like in the beginning, right? As a little baby. You sure don't look like that now, right? You have developed, okay? You went through childhood development, adolescent development, reproductive development. Yeah, your reproductive organs were non-functional at the beginning, and now look at them, okay? Now they look different, they function differently, right? So changes happen during the lifetime of an organism. You know, and this is, again, characteristic of a mushroom. You know, you go out one day, it looks different, you know, a week later. You know, plants and animals and all kinds of living things change during the course of their lifetime. You know, non-living things like a rock, you know, very little change, right? Now, there could be like that wind and water take a toll and the shape of that rock changes, you know, due to erosion and so forth. One thing I want to point out about these things that we're talking about, like development changes could happen to non-living things. How about movement? My car can move, but my car's not alive, right? So non-living things can have these characteristics as well, but they don't have all of them. Living things will, every living thing, a bacteria, an amoeba, a plant, a mushroom, you know, fungi, humans, animals, have every single characteristic that I'm talking about. So that's kind of the definition. Some non-living things can do some of these things, but, you know, my car can't grow. It doesn't get any bigger. Okay. So, what else do we say? So the car can move, but it can't grow. Changing structure. A rock can change maybe its form if the wind and the water take, erosion off of it or something, deposits on it, but it doesn't move. There's just really not really any movement or irritability. It's not responding. Rock doesn't change its temperature or anything like that. So, all right, what else do we have? Reproduction. All living things, according to their natural state, have some ability for production of new organisms. Okay, so it can create more of itself, more of its kind. Metabolic processes happen in all living things. And I've got a whole list here. Just checking my notes here, a whole list of different things. I don't know if you'll have a chance to write all these down. If I go too quickly, pause the video, right? Or go back to your textbook and look. And I do want you to write these things down. And if you need to, write down definitions. Do you know what each of these mean? Like respiration. That one's one, you're like, oh yeah, that's breathing, right? Actually not. Breathing is ventilation. You ventilate. You bring air into your lungs and you exhale the air out. Respiration goes a little further than that. It is an exchange of gases. So you're going to take oxygen into your blood and you're going to release carbon dioxide from your blood. That's respiratory process. Why do you take oxygen in your blood? The ultimate goal is of being able to convert energy. In our bodies, we take the oxygen in because we need that oxygen, for example, to convert food energy into a molecular energy for cells. And eventually we'll talk about this. That cellular molecular energy is called ATP. Maybe you've heard of it. Okay, so they might give a little bit of definition. Check that out in chapter 1, okay? If you're not, read through that and make sure you have as much understanding as you can about these metabolic processes and what they are. Like digestion, yeah, okay, I know that's when I eat stuff and it gets broken down, right? See what else your textbook says in chapter 1. We have next semester in 102 a whole chapter on digestion, a big chapter at that. So there's a lot more we're going to learn, but learn as much as you can right now so that at least you have a good idea of digestion, respiration, and like, what about absorption? What exactly is that? Is that different than digestion? Yes, it is. So find out a little bit more about these definitions. Okay, check those out in chapter 1. Another critical characteristic of living things is the composition of cells. It's made of cells, okay? All living things have one or more cells. Your body has cells, right? It's made of cells, trillions of them actually. We're pretty sure that you have like over 1 trillion cells. Trillions of cells make up your body. A bacteria is just one tiny little cell. That's the whole bacteria. So it can be a few cells, 100 cells, hundreds of cells, thousands or trillions of cells like in the human body. A big blue whale is going to have many trillions of cells. We will talk more about this in chapter 3. A whole chapter on cells. Okay, so we'll look at that much more in depth in chapter 3, but that is critical. A rock, you will not find a cell there unless it's something living that's living in or on the rock, okay? But the rock itself has crystalline structure perhaps, but not a cellular structure, okay? So that's a key difference between living things and non-living things, the presence of a cell. Oh, and I wanted to mention another key point about that. This allows organization. That's a key feature of living things. That, I mean, crystals can be organized too in a crystalline structure, but this is a unique kind of organization. And the chapter 1 will talk about that organization. All right, and one more key characteristic, maintaining homeostasis. We have a whole section in the end of the chapter about that. This is talking about basically a stable state of being. So, for example, being able to maintain our body temperature, right? Our body temperature, we don't get freezing cold and super hot. Like if you did, you would die, right? If you got too hot or too cold. Our temperature might fluctuate, but we maintain a relatively constant, stable state of temperature. That's our homeostasis for body temperature. Blood pressure, blood sugar, things like that, you know, generally remain pretty stable. So this is a key idea, which we will talk about here again at the end of the chapter. So important in healthcare, because the whole goal typically in healthcare is to look at those parameters like temperature and blood pressure and so forth, and make sure that we are maintaining those, because if you don't, you might die, right? Characteristics of living things have some stability. Now, again, this varies, you know, like certain lizards, they let their body temperature get really high or very low, and they can survive that, but there's still limits, right? You can't take a lizard, okay, to the North Pole. They can't survive it. Likewise, some polar bears can't survive, you know, at the equator, right? It's just too hot for them. So we're adapted to certain environments, but there are limits, you know, and we have to stay in that stable state. Now, a rock, you can take a rock from the North Pole and put it at the equator, and it doesn't hurt the rock, okay? They don't care. They don't mind about changing states so much. But living things, it's really important to maintain homeostasis. And living things, our bodies are designed to try to survive, okay, and keep it a certain way. So if you get too hot, you sweat. If you get too cold, right, you shiver. So we have processes in our body to help us maintain homeostasis. And hopefully we can do that, right, to maintain life. Okay, moving on. All right, so now we have a little better idea of, you know, what we're studying as biologists, okay? We're studying living things, and we know how to identify and what we're looking for and trying to monitor, in a sense, in living things. Now, specifically, though, we're studying anatomy and physiology, okay, in our biological studies. Now, what does that mean? So anatomy, we're studying the form. Another good word here might be the structure of a living thing, okay, or how it's built. Okay, that's anatomy. That's what we're doing in anatomy. We're looking at how it's put together, what's its shape, what's its size, how is it designed, so forth. Some examples that we'll be doing in this class. Okay, here's a picture of a sheep's brain. Toward the end of the semester, we would like to do a dissection of a sheep's brain. Now, we're currently in the pandemic and trying to figure out with virtual classes how we're going to do that, but we're going to try. We might be able to send some things home with you. So we'll work on that. This is a sheep's brain, and in lab class, at the end of the semester, we always dissect a sheep's brain. And part of that dissection is identifying the parts, like this line right down the middle is called the longitudinal fissure. This like lump back here at the back is called the cerebellum. So we name the parts and we see, oh, this part is separate from this part, and these two parts of the cerebrum are separated by a fissure. Okay, so that's anatomy. We're here, these pelvic bones. We're going to name parts of the bone. We're going to name the bones and identify little bumps and nooks and crannies and holes and so forth. We're going to do that in this class too. We're going to study bones. We're going to look at pictures of bones. If you can get onto campus, we're going to have like actual models of a skeleton, and you get to touch them and feel them and see all those things. So that's anatomy. We do a lot of this in the lab because we have models and we have specimens and stuff. So mostly in lab, a lot of the labs we're going to be doing are anatomy labs. What we're doing right here with the dissection or looking at bones is called gross anatomy. Okay, not because it's disgusting, right, dissecting, but because it's large. Gross means large in this case. And it's large enough you don't need a microscope, okay, to see it. When you can see it with your naked eye, or with your glasses, that's gross anatomy. Okay. We will also do some microscopic anatomy where we'll get like maybe a magnifying glass or a microscope, okay. More microscopic anatomy is when you really need a magnifying lens to be able to see something. So, I could take a magnifying lens and look at my skin. That's bordering on microscopic, but usually a microscope, something that's even smaller that you need more than a magnifying lens. So, and this pink and purple stuff. This is dye was used, like pink and purple dye, to color a little piece of skin. And we put that under the lenses right in here. And we look at the skin under the microscope, and looking through those lenses, those magnifying lenses, this is what you would see. Lots of little circular shapes. Those are the cells. These little bubble-like structures are the cells that make up your skin. Can't really see that. I can put a magnifying glass. I can see little hairs and bumps and ridges on my skin. I can't see the cells. So at that level, cellular level, we need microscopes because you can't see cells with the naked eye. You have to magnify them even larger. I can see like bumps and ridges, but I can't see the little bubble-like structures that are part of my tissue of my skin, right? So we'll do some microscopic anatomy in this class too. And we're gonna like see, are they, some of these look more flat, and some of them are more rounded, and some of them have dark spots, and some look empty. That's structure. How's it built? Is it big? Is it tall? Is it skinny? Is it layers, right? Is it dark staining or light staining? We're gonna look at those different characteristics of how it's structured and put together as part of our lab activities. Okay, where we do a lot of anatomy studies. We're going to name those tissues and name these bones and name the parts. Anatomy involves a lot of memorization. Okay, so if you're good at memorizing new words, which is kind of like learning a language. So part of this class, if you ever took Spanish class or German class or something, it's a little bit like that. You have to learn how to talk like a scientist. And that was going to learn a lot of new words. Okay, so as we're doing our studies, we have the right words to describe, both the shapes and what we just call those parts. You can talk to people about them. And new words too, like not belly button, but umbilicus. Okay, so lots of words. In that first lab activity, we'll introduce a whole lot of anatomical terminologies, lab 1. Okay, dive right into it and start finding out some of these basic vocabulary words that you need. That's how you'd start like a German class or Spanish class, right? Start memorizing words. How did you learn a language? How did you learn the first language that you speak? Maybe English or Spanish or Russian is your first language. How did you learn that as a little kid? You just listen to people. So you want to be watching videos. You want to be listening to me talk. You want to get on YouTube and listen to other people. Watch videos on your Master A&P, watch videos and listen to people talking about stuff. Here are the words, and then you use them, right? As a little kid, you like repeated everything that your big brother said because it was cool, right? Repeat after me, right? Say these words, practice them. That's how you learn a language, right? Listen to it, practice it every day. Think about them a lot and keep using those words so that they become part of your new language. Okay, and we are also studying physiology, and physiology is a study of function. I might also use the words here. Let's see, what was the other good words? Oh, process. Processes, okay? Processes of the body. You're gonna need to learn the steps of an activity in the body, for example. Okay, this is physiology. How, this is the how, okay? How something functions, how something works or how it functions. That's physiology, And we're going to be doing that too. We're going to do a lot more of that by reading in your lecture book, looking at diagrams. So this is how a bone grows. Like in lab, okay, we'll just see the model and we'll name, okay, it's a little bump here and a ridge here, and this bone is called this, and this is its shape, and this is how it connects together. That's anatomy. But in the lecture book, we're going to see how like a baby's bone is different from an adult's bone. What has changed? How has it grown? How has it developed? That would be another thing, development. How has it changed? But how did it get bigger? Right? Start out like little bones and then we get big bones. How did that happen? Okay, that's what this diagram is explaining, how that happened, how that bone got to be bigger, okay, through a process of ossification. Okay, so studying that process. First this step, then up here it starts, and then later this happens. So step by step, seeing how that bone got bigger. Or here's another one. So on a microscopic level, these are the cells that make up your nerves and in your brain. They're called neuron cells. So they're kind of rounded. I see over here in the skin, you know, these are rounded cells. This one's kind of rounded and it has a dark spotted nucleus in the middle, but then it has branches, lots and lots of branches, because the neurons are responsible for picking up signals and then sending those signals. So it's kind of like lots of little fingers out there to receive, and then a long branch called an axon, because once it receives information, it may have to send that information somewhere else in the body and communicate. That's what neurons do, communicate to other cells in the body to tell them, hey, something's going on over here. You need to do this. Hey, I found out we're getting too warm. Let's reach out to those sweat glands and tell them they need to make some sweat, right? How do they communicate from cell to cell? So here's one neuron communicating to another. I can't see this. I probably can't even see this very well with our microscopes. You need a really super powerful microscope to see these tiny little details of a cell. So sometimes I can't see it, even with the tools that we have. We maybe need, you know, bigger, more powerful microscopes, or sometimes like, you know, some of the chemistry and physics, you know, of these tiny little molecules, we can't always see it, but we make drawings of it. Like if we could see something that tiny, this is what we see. So in this red circle, it's magnified here to this big red circle and saying, okay, the ends of those little branches, there are the yellow dots here are coming out of the one and going to the other. These are molecules called neurotransmitters. So some chemical molecules are coming out of one neuron's branch and going to another neuron's branch and getting stuck on the receptors so that it's picking up those chemicals. And that causes a change in this cell. So now that cell's like, oh, okay, I have these different chemicals. I'm going to do something different, right? I'm going to take that stimulus and now I'm going to react and, you know, do something different, like go tell that sweat's, you know, gland that it needs to produce sweat. So we're learning how does a neuron send a signal? That's physiology, study of function. How did that work? Okay. First, I start with the anatomy. Okay, it's got branches, and I learned the names. These are dendrite branches. This is axon branch. So the anatomy is good to know what the parts are called, but then how do those parts work? Okay, figuring out how they work. So a lot of times, what some universities do is you will take anatomy class first for one whole semester, usually like a ton of time in the laboratory, just every day, like, here's this part, this is called this is called it. And you just do all the anatomy first. Okay, lots of memorization. And then the next semester you go back and say, okay, remember those neuron cells, remember those bones, this is how they work. At Ivy Tech, we've decided to put the two together. Okay, like while we're seeing the bones in the lab, we're going to learn how they work, how they grow. While we're looking at the brain, then we're also going to look at the neurons that are in the brain and see how they send signals. We do anatomy and physiology together. And I like that because they're related. They're absolutely related. So, you know, I can't get to the whole body in one semester, so we still have 2 semesters. Like most universities do, like a semester of anatomy and then a separate semester of physiology, and they got 2 semesters. We're going to do it in 2 semesters, but we're going to do like part by part. We're going to do like the skeletal system in lab and in the lecture, we're going to learn the physiology, and then we'll move on to like the muscular system and look at the muscles in the lab, but then figure out how they work, how they contract in the lecture. So we put the two together, okay, in this sequence. And the reason for that is because they're absolutely related. So this is one of the key principles or core principles that your textbook author outlines at the end of the chapter. She said, here's a key thing that you need to know about studying anatomy and physiology in our bodies, is the fact that structure determines function. Now, just a little side note here, Ammerman says it different. That's the textbook author. She says form, see how she says it, form follows function. Okay, that's what she's saying. That's a little different. That's saying that form follows. That's kind of saying like the structure follows because of the function. I think that's backwards. Just as a biologist, I want to disagree with that, but I'm not going to get an argument with her about it right now. Same idea. Let's just say that they're related. Okay, now whether one causes the other, I think structure causes the function. The way something's built causes how it's going to work. It's not like, oh, I want this to work, so I make it this way. Our bodies aren't really like that. We just appeared with our bodies, right? We can get through it. Take my biology class, right? We can talk about the evolution and development of humans and how we got here, right? But my take on it is that you were created, okay? You were created through an evolutionary process. I believe in both, right? Creation and evolution. You were created through an evolutionary process, and you have a certain structure. And based on how you're put together, what your structure is, that determines how your body works. Okay, as long as we agree that they are related, right? So structure, anatomy, and physiology function are definitely related. In fact, I'd say that the anatomy determines what the physiology is going to be. So it makes sense that we're studying them together, because once we know the anatomy, that gives us a clue. Like, oh, okay, I know how it's built. Now I know how it works. Sometimes just seeing how it's put together gives you a clue how it works. Look, for example, at this screw and a screwdriver, right? And you've seen these before, right? Oh, that little cross head there fits in the little cross hole there. Okay, so if I put it in there, it sticks in those little notches, and then, okay, I have a handle, I can turn the screwdriver, and that's going to help, turn the screw and put it into place. That's the structure. You see how, and then we figure out, okay, that's how that works to turn the screw, right? But it has to be, structure is very important. If it didn't have the right structure, like right here, look at the screwdriver with this screw, you're not going to get the function that you want there, right? And so structure is important in determining how something works. If the screw doesn't have that cross shape in it, this screwdriver doesn't work with that. It's just not going to happen. Right? So that's really, really important in healthcare. And that's the approach sometimes we get. Like, often a patient comes in and they're like, doctor, this isn't working right? Like, my blood pressure is always too high, or it's too low, like it's not regulating right. Okay, well, sometimes we go in and look at the structure, like, are your arteries clogged up with too many fats, right? They might do a scan of the blood vessels in your body. They might do an ultrasound or a heart scan or something. Look at the blood vessels and say, like, okay, are the tubes wide open so the blood can flow through or not? Or are they getting clogged up? So sometimes it's a matter of, like, taking an image, taking an x-ray, taking an MRI, and looking at the anatomy, or getting a scope, like looking at a scope. You put a scope down through somebody's esophagus, right? Look at their stomach and see what's happening in there, right? Are all the little valves and the folds in the right way, to allow the food to be digested. So you might look at the structure to see if something's not functioning correctly, right? So very important concepts here. The structure has to be right to determine the physiology. Here's another example, anatomy determining physiology. The blood flowing through these little blood vessels here. We're seeing red blood cells. Notice how the shape of those what the shape of those red blood cells are. They're small and rounded, kind of hollow in the middle so that, they can flex a little bit. That helps them to get through really tiny capillary blood vessels of our body. Sometimes they have to kind of line up one by one and squeeze their way through there because some of the blood vessels are very tiny. So the cells need to be very tiny. These cells are carrying oxygen. We need oxygen, we said, for respiration. Part of respiratory process is oxygen has to come into our cells so we convert food energy into ATP energy. These blood cells got to get through. They got to travel and get to all the cells of your body to carry the oxygen. What if they couldn't get through the tubes? I mentioned about fats like clogging up the tubes. That could be a problem. Or in some cases, it's the red blood cells themselves that don't have the right shape in a disease called sickle cell anemia. Instead of the red blood cells being rounded and having smooth edges, the red blood cells are elongated and they have pointed edges. And those pointed edges can get caught on each other and they can get caught on the walls of the blood vessel and they can create a logjam and block the flow. This is a sickle cell anemia crisis. The person's not getting blood flow to certain organs. Very painful and very deleterious, right? Can damage the organ. It could lead to death, right? So very important that we keep their blood flowing. Sickle cell anemia, it's a genetic condition where the DNA instructions are wrong and the person's instructions for how to make their blood cells isn't right. And they have these oddly shaped blood cells. Coincidentally, some exciting research in sickle cell anemia. I've been reading this past year how we've found some treatments that are actually very effective at changing the person's DNA instructions. Wow, like genetic reprogramming in a sense that have actually worked in some individuals. So very exciting stuff. Okay, so that's when you can see the difference in the red blood cells. Sometimes it's something you can't see. This is talking about another disease called phenylketonuria, or the short way of saying that is PKU disease, phenylketonuria disease. In this condition, it's also a genetic condition. The person's enzyme called PAH enzyme is not working. And in relatively recent years, we found out why. The enzyme doesn't have the right shape. The enzyme has to have the right shape to interact with a certain amino acid. Now, what's an amino acid? We're going to get into this in the next chapter. Chapter 2, we'll look at proteins and how they're built of amino acids. So this is a protein that you eat, and your body needs to break it down into individual amino acids. Each amino acid can do different things in your body, and your body needs to be able to digest some of those amino acids. But in a person with PKU, their enzyme for breaking down phenylalanine. So here, normally, this phenylalanine, this purple dot, and it's just a purple dot, right? And these amino acids are different dots because these are molecules that are so small, I can't even see them with a microscope. So we just drew dots. These small molecules get broken down, like see you're broken in half. They normally get broken down by an enzyme in our body, but in a person with PKU, they have a defective PAH enzyme, so the phenylalanine builds up in their body and becomes toxic. So interestingly, we've just discovered, and again, in the last 5-10 years, I think we've discovered the shape of that enzyme. And again, we just draw little blobs and dots because I can't really see a molecule. We don't have microscopes that are that powerful. But we do know that they are, this protein and this enzyme is also a protein, have strings. You can see the beads on the strings of amino acids. So that's what this is trying to show us, the strings and the strands of beads here and the strands of coils here. These are different molecules that are strung together and create a specific three-dimensional shape. I'm just trying to show us the three-dimensional shape. It's kind of exciting research in PKU too, because we most recently identified the shape of that enzyme, and we've identified in PKU what's different about the shape. And the fact that they don't have the right shape, that amino acid, phenylalanine amino acid, can't hook up with this enzyme. So phenylalanine just never gets broken down because the enzyme doesn't have the right shape. So again, even down to our little molecules, our molecules have to have the right shape, the right structure, so that they can function correctly. So on a larger level with blood vessels, or a small level with molecules, shape determines, structure determines function. Ammerman goes into some other examples about, here, the lining of the blood, the lining of the lungs is normally very thin, but in some conditions, like fibrosis of the lungs, extra cells get built up in the lining of the lungs, and it's just too thick, and the gases can't get through to the blood or from the blood. The carbon dioxide can't get out either because there's too many layers there. So, again, the structure, the linings in our lungs have to be thin so that when the air comes in, it can get through those thin layers to get to the blood. So structure determines the function. Okay. So examples of structure determining functions. Speaking of structure, one part in the chapter, they introduce the idea of organization. I mentioned that's important to humans, so cell organization, but even more than that, the organization in our structure is important. And while we're talking about structure, what are we made of? Okay, our structure has to be put together in different, what are the pieces that we are made of? That's an important question to understand in this first chapter. Okay, what are we talking about there? Well, I've showed you some things. Well, there were blood vessels, and there were bones, and there were neurons. And then there were these enzymes, and there were proteins. But I said we're made of cells. Is your head spinning? What is your body made of? Well, I have skin and a brain, and right, there's a lot of different answers that you can give here. I was being silly here with this word art here. What are you made of? There's one answer you could say, like sugar and spice and everything nice. That's what little girls are made of, right? Snips and snails and puppy dog tails. That's what little boys are made of. What are we made of? Just a side note here. I put this in just for some levity, okay? But please don't take offense to this, guys that are out there in the audience, because in our house, we love puppy dog tails, and we think snails are very, very cool, okay? And I've been known to collect many, many snippets of many things, so I'm quite the collector. So don't be offended. I love these things, and I love these things. You're all very welcome here. So, but seriously, guys, what are we made of? How would you have answered that question? You might have said, oh, well, I have cells, right? Or you might say, well, I have a brain, and I have skin, and I have bones. But maybe first thought it came to you was like atoms. I'm made of atoms. Guess what? You're all right. These are all true. What are we made of? You are made of organs. You are made of cells. You are made of atoms. What does all that mean? It's just different levels. Remember, we're getting at here. Organization. And there's like, how are those atoms and how are those organs and how are those cells like put together, and different levels. So what I want to get to, we're going to look at levels. Here's one more example just to like help you see the difference. I'm wearing a sweater. See if you can see this sweater. What's a sweater made of? It's a fabric, right? It's sweater fabric. Or you might, if you know a little bit about sweater making, right? There's like yarn, right? And the yarn is like knitted together in here. Like I see white and gray and stuff. It's knitted together. So it's a fabric, but if you look closer, it's actually yarn. Did you ever look closely at yarn? Yarn is a bunch of little threads, right? All interwoven together. So yeah, you could say it's fabric, or you could say it's yarn, or you could say it's threads. What's a thread? If you look at a thread is a lot of little tiny fibers. Well, what are those fibers? Cotton or wool. So, like you could look like closer and closer and closer. And that's really what we're talking about here too. Like look at the big scale here, like this person, this whole organism. What are they made of? Well, they have a digestive system, and they have a respiratory system, and they have a nervous system. Or you could look at the organs. Well, they have an esophagus and a stomach and a liver. Those are the organs of the digestive system. But we can look at that esophagus and say, well, that tube is made of certain tissues. Like tube is primarily stratified squamous epithelial tissue. It also has smooth muscle tissue in it too. Your stomach also has smooth muscle. There's a lot of muscle tissue in the digestive system too, so it can squeeze the food along through the tubes. There's different tissues that make up the esophagus and tissues that make up the stomach and the intestines. So if you just look closer and smaller, just like my sweater, if you look closer, well, it's actually yarn. Well, those yarn are actually threads, and those threads are actually fibers, and those fibers are actually molecules, right? You get down to molecules. In our bodies, like I said, when we got the microscope out, we saw the little bubbles, and these are kind of elongated bubbles. These are the cells. We can look at the individual cells in that tissue. So I need a microscope to see it, but my body, these organs made of tissues are made of cells. I can see them with a microscope. Now, what I can't see with a microscope, because even our really, really powerful microscopes have trouble seeing, are the chemicals. They're just so small. So that's where we start just totally like putting shapes, like little balls and squiggles and triangles and squares and stuff, because we don't really know exactly what their shapes are. Okay. Chemists and physicists try to look at that stuff. Okay. Now we're going to get a little bit of that. You might notice that chapter 2 is about chemistry. We're going to do a little bit of that. This is not chemistry class, but we got to understand a little bit about what we're made of. Our cells are made of chemicals like proteins and carbohydrates and things like glucose and water and salts and what else? ATP molecules and phospholipid molecules, like it says here. So there's a lot of different chemical molecules, and those molecules, we'll find out, are made of atoms. So we're going to find out atoms. That's what like physicists study, atoms, right? We're going to do a little bit of that. Okay, we've got to have a little bit of understanding of what an atom is and what a chemical is, so that we understand a little bit about what our cells are made of. And that's where we're going to spend more time though, is in getting into like looking at those cells and just trusting, okay, the chemist told me it's this, and the physicist said it's shaped like this. They can't even see them. They don't even have microscopes to be able to see them, but they do other fancy lab work to be able to understand like how these are shaped. So they do kind of the anatomy of an atom, the anatomy of a molecule. They look at the shapes of molecules, just like we were looking at those balls on a string for the protein and how it was bent and coiled and all that. That's what chemists do, a lot of that, and physicists some of that too. But they look at the shapes. So they do some anatomy kind of stuff, but on a really small scale, right? And anatomy and physiology of the human body, we need to know a little bit of that, but just so we can see the shapes of the molecules making the shapes of cells. Because these shapes of the cells is depending on what they're made of. Just like the yarn is threads, but what are threads? Threads are like fibers, right? Long, skinny little strands. Like, how did they get that way? Let's look at the actual molecules that are long and skinny molecules that make up fibers. Okay. So something to think about. And there's a tutorial, I think when you put the PowerPoint on your computer, you can click here and watch a little video about that, or go to Mastering A&P in the study area. And she talks about in her book, getting the big picture of how our body is put together and how it's organized. And it is very organized. We have to organize our molecules into these bubbles called cells. And then those cells organize themselves in, like, you can see like rows and layers and how they organize. We'll see the organization into organs. But then we look at how the organs are connected. Like this one has to go from the mouth to the stomach, and then the stomach has to connect to the small intestine. So we look at how they're organized in an organ system, and then we see the different organs working together. And then once we have a digestive system and a respiratory system, we look at how those systems work together in the organism, the whole thing. So we're going to look at those connections and Yeah, and we're going to do it kind of incrementally like that. You'll notice chapter 2 is chemistry. Chapter 3 was about cells. Chapter 4 is titled tissues or histology, the study of tissues. And then we get into organs and organ systems. So this kind of guides our study too. We'll spend the first half of this semester looking at smaller things and see how the smaller things can put together to make bigger organs and part of the organs systems of the body. So kind of levels of, what are we made of? We're going to start small and work our way up to big. Okay, last part of the chapter, Ammerman outlines some core principles. Okay, looking in your notes. Okay, there's a module. We just finished the second module, module 1.3. I'm skipping over that because you'll totally cover it with your lab work. Okay, language of ATP, of A and P, the language of A and P, anatomical terms. You're going to learn that in lab, and actually module 4 as well. Organizing human body. Let me just real quick direct you back. Sorry, thought it was going to be quick. Yeah, let me just pull up again, just make sure everybody's on track. In your notes, we just did 2. Here's 3, the language of A and P. Module 4, organization. You're going to cover that in lab. So I'm going to jump to the end of the chapter, module 1.5, core principles. Ammerman identifies 4 key principles. We're going to spend a good bit of time on homeostasis and then real quickly do the last 3 principles, key ideas that you need to understand. And we will work on a lot throughout this semester and next semester. All right, so let's talk about that first principle, the idea that we have homeostasis. And we mentioned that that's a key characteristic of living things. We have a relatively stable environment, like our body temperature, our blood pressure, our blood sugar level, salts and ions. Like you shouldn't have too much sodium in your blood. You need to have enough calcium in your blood. All these things have to be regulated and maintain the right amounts. Hormones, sometimes you make more hormones, sometimes you make less, but it needs to be at a right amount. There is a right amount, right? And we usually stay kind of, it might go up a little, might go down a little, but we usually have a certain range that our temperature, our blood pressure, our hormone levels need to be in that range, right? For that stability. This word homeostasis literally means same state, state of being, standing. You're standing at this position and you need to stay at that position, right? Homeo means same. The words like homosexual is the same origin of the word, means same sexual interest. Stasis, static, stat, right? Stat, when they yell stat in the hospital, like, whoa, everybody stop what you're doing, like stop a minute, stand still and look, right? Because stat means, or static, like it's just same noise over and over and over again. And you're not hearing the music, right? You got static. So kind of that stasis the word comes from the same word roots there. When you see it, it means that same static state that we're trying to maintain, homeostasis. How do we do that? Well, we're going to get a lot of details as we go into different chapters of the book, but generally homeostatic mechanisms, there are processes to maintain homeostasis. We call them mechanisms. And those mechanisms always have 3 different parts of the body. Well, sometimes one part can do 2 jobs, but those 3 different parameters have to be covered. Sometimes it's in the same part of the body, sometimes different parts of the body. And they have to, if they're different parts, they have to work together to do 3 things. There's kind of 3 steps to the process. Okay, so the first part, the receptor, it's also known as a sensor, has to detect the stimulus. You're not going to do anything. You're not going to change anything. You're not going to respond if there's no need to, right? If it's not broken, don't fix it. So first you have to know if there is a reason to adjust, right? We always want to adjust and get stable. But if you are stable, you don't do anything, right? But if there's a change, first you have to detect the change. So that's what a receptor does. It is constantly monitoring. You may call this the monitor, the sensor, the watcher, okay? The receptor, and in other words, sounds like receptor is receiver, receiving information. Think of like a wide receiver, receive the football, right? You receive something. A receptor receives information. It's always like measuring, detecting what's going on around you. And if there's a change, that change is a stimulus. And the stimulus will initiate an additional change by the body. And it does that by the receptor, like detects the change and sends the information. Okay, let's maybe say it that way. Sends a stimulus. The receptor sends information to the control center. Okay, the control center, also known as an integration center, processes the information. So some keywords here. Detects first, you just measure, pick up the information, and then you process that information. The receptor can't process. The receptor has no idea what to do. It's just like, hey, something's going on. Okay, I detected it, but now the control center has to figure it out. Like, what does that mean? It processes the information by comparing it to a set point. Well, the measurement said this. This is where I want to be. Is it good? Is it not good? Right? You got to compare. Like, what should my body be? The control center, in a sense, has a programming. It knows what you should be, and that's the set point, the desired value. And if the receptor is saying, well, here's where we're at, and the control says, well, here's where we want to be, and you're saying we're here, oh, that's not good. The control center processes, and the control center can make a decision too. That's an important point. Okay, if a response is needed, okay, the control center can decide on the appropriate response. Control centers are very often in your brain. Not always, there's some other places too, but a lot of times control centers are in your brain. Okay, that's where we process information, right, and make decisions. And the control center will send a command. So it figures out what to do, but it doesn't do it. Your brain doesn't like do it. Your brain just tells the other part of your body, hey, do this, right? Sweat glands, you need to sweat. The neurons send a signal, right, to an effector. So that's the last part. So part number one, the receptor detects, the control center processes, the effector responds, okay? And effectors are almost always, I can't think of any exceptions, there might be, but almost always, maybe always, a muscle or a gland. Like muscle cells or gland cells, like we mentioned, sweat glands. If I'm too hot, right, get my sweat glands to sweat. They're doing the work, okay? Creates a change. These guys are actually doing it to cool you off, right? They're doing the sweating to cool your body off. They actually do something to make it better, right? To fix the problem. Now, sweat glands don't know when they're supposed to sweat. You don't want to sweat all the time. They don't get to decide. They are under control of the control center. The control center will send a message when they should sweat and when they should stop sweating. All right, so you got one that can measure, see where you're at, one that can process that information and make a decision and send the command, and then another part of the body, usually a muscle or gland, that actually does the work to correct whatever the change was that we needed to respond to. All right, here's an example. So we've been talking about body temperature, and here's a diagram. This is from another textbook, homeostatic control of body temperature. It says start right up here. Let's follow this kind of in order. Homeostasis is disturbed. Let's say, okay, your normal body temperature should be about 37 degrees Celsius or 98.6 degrees Fahrenheit. But let's say you get too warm, okay? The heat's too high or it's hot outside, you go out on a really hot day, and all that heat is being absorbed in your body and your body temperature rises. You have temperature receptors in your skin and some in your brain too. These are they're actually neurons. Okay, these are neurons called thermoreceptor neurons. You have neurons in your eye that are called, sorry, let me think of the right word, photoreceptor neurons, okay? Photoreceptor neurons, or let's see, trying to think of some other neurons. In your tongue, when you taste something, they're chemoreceptor neurons, chemoreceptor. Thermoreceptor neurons can detect temperature. Different neurons can detect different things. In your tongue, they can detect chemicals. In your eyes, they can detect light, photoreceptor. That's what they're called, neurons. Thermoreceptor neurons are neurons in your skin and in your brain and other parts of your body too that can detect temperature changes. If it starts getting warmer, those neurons that are the thermoreceptors, okay, send a signal. The neurons send a chemical signal. They're connected to the brain, specifically an area of your brain called the hypothalamus. I'm not going to test you on this in chapter 1, but we will learn this when we study the brain, and you have to know where your hypothalamus is and what it does. It's kind of in the middle of your brain where this purple square is. And one of the jobs of the hypothalamus is to regulate body temperature. It receives input from the receptors, and it has like a programming that it knows, like, oh, that signal is saying I'm this temperature, and I know I'm supposed to be this temperature, so that's too hot. Okay, so I got to do something about it. So when that signal comes in from the receptor saying this is the temperature, and it's like, nope, that's not what I wanted to hear, I better send a signal to the sweat glands. Okay, so then this arrow is showing it's sending information. The neurons are connected to, for example, the sweat glands. And it tells the sweat glands, start making this watery stuff and put it out on the skin, and that's going to evaporate and cool this off. It has additional neurons connected to muscle around your blood vessels in the skin. And those blood vessels in the skin the muscles relax, and the blood vessels like have more room, and they open up, and more blood starts flowing to your skin. And what you get all red, right? Because all that blood flowing in your skin. But that's like a radiator, right? All that heat from that blood flowing in your skin, the blood has a lot of heat in it. It's right there on the surface, near the surface of your body. It can radiate that heat outward and help cool the body. And the evaporation of the sweat cools the body. Notice we're looking at muscles and glands made changes, right? The muscles relaxed, the glands started producing sweat. These are effectors. They have an effect. They created an effect. So that effect allowed our body to be cooler. We're back at a good body temperature. So homeostasis was restored, okay, through the action of that homeostatic mechanism. Now, there's another arrow here. Notice that if we're at normal body temperature, if there is no change, okay, if the body temperature is not high or low, then the neurons are not stimulated. If you have no light in your eyes, neurons are not stimulated. If you have no chemicals in your mouth, you're not stimulating. If there's no temperature change, then there's no stimulation. If there's no stimulation, then there's no signal, right? Cross this out, no signal to the control center, and there's no need to send a signal. And then we just stay stable, right? Stay in homeostasis. It's just if there is a change, then that change stimulates receptors, and the receptors stimulate the control center. The control center will figure out what change I need to make. It's not always your sweat glands. What if the neurons detected cold? If they detect cold and send to the hypothalamus, then something else is going to happen. I think we have that on another slide here. Notice receptors in the skin. This is a different picture, but receptors in the skin detect the change. Let's say it's getting too cold, below normal range, then it sends signals to muscles on your bones called skeletal muscles. And you start to shiver and shake, right? Your bones, your skeleton starts moving because your skeletal muscles now are contracting rhythmically. It's not that shaking is good for you, but the contraction of skeletal muscles happens to create heat, okay? We'll learn about that when we study muscles. When your muscles contract, they move your bones, but it also makes heat for your body. So that's going to be a good thing to warm your body back up. So this is Ammerman's picture from her book. Notice that she showed it like a needle, right? That, like you want the needle on your gas gauge, right? Or your temperature gauge on your car to not be too hot, not be too cold in your car. Same thing here. You don't want it to be your needle on your control center, right? Your set point for your needle. You don't want it to go too high. You don't want that needle to drop too low. You want the set point in your hypothalamus says, it should be this, right? We don't want to get too high. Don't want to get too low. The receptors are detecting highs and lows. It's going to trigger information to be sent to the control center. Control center decides what to do about it and sends the right command to the right part of the body, either skeletal muscles or smooth muscles or sweat glands to respond appropriately. Okay, so that's the parts of a homeostatic mechanism. We saw one was like a circle diagram. This is a circle, but it has a needle, like don't get too hot, too low. There's other ways. Some textbooks show a teeter-totter. That's a good idea. Like, can you picture that? Like, I don't want to get too hot. I don't want to get too cold. My teeter-totter should all be balanced. Right? So that's a way to think about homeostatic mechanisms. Think about a teeter-totter. What's going to happen if it goes too far this way? I might sweat. What if it goes too far this way? I might get shivers, right? To get it balanced out. Keep your teeter-totter balanced. That's homeostasis, when you're in good balance. Sometimes textbooks use this, a figure 8. Figure 8 is nice because homeostasis is right in the middle, like this showing blood glucose levels should be this. 70 to 110 milligrams of glucose per deciliter of blood. That's what you want. Now, it doesn't have to be exactly 70 or 80 or 90 or 100. It can be anywhere in that range, but that's the range. If it gets higher than that range, there are beta cells in your pancreas that detect the high levels and they make insulin. That's your effector. And then what happens? Because the insulin, these are things that change in your body to get your blood glucose back to normal. What if your blood sugar gets too low? Again, there's cells in the pancreas, but it's the alpha cells this time that detect. That's your receptor. And the kind of a control center to make glucagon. I guess you could say glucagon is a hormone that creates an effect. That's the effector, glucagon. And this is what happens, and it gets your blood sugar back up. Puts more sugar in the blood, so you get back to homeostasis. So just another example, we'll study this when we study hormones like insulin and glucagon hormones. We'll study that with the endocrine system in Anatomy 102. But another example, and I like that figure 8. Like, don't go too high up, don't go too far down low, right in the middle, right? Things get higher, bring it back. Things get lower, bring it back up. Stay in the middle. It's about balance. Somewhere in there, okay. Yeah, it can go, this is body temperature. It can go up a little, like you notice, this is normal body temperature throughout the day, like 7 a.m., 10 a.m., afternoon. Your body temperature might change throughout the day. Like when you're sleeping, it might get lower. It might go up, but it shouldn't really get above 99. It shouldn't really get much below 97. It hovers around 98.6 or around 37. There can be some variations, but we don't want to get too high, you start to bring it back down, or too low, you start to bring it back up so it doesn't go too far out of whack. Now, what was mentioned on some of these, yeah, they mention in here, Ammerman mentions, and I know the previous slide too mentioned, a negative feedback loop. There's a loop here, and we can kind of see that. I like that with this figure 8 too, the loops. Loop up or loop down. What are these loops? Well, there are two types of loops. Core principle, feedback loops are a key part of homeostatic mechanisms. And there are two different types of feedback loops. So all homeostatic mechanisms, we all have mechanisms for our blood sugar, we have mechanisms for our temperature, we have mechanisms for our blood pressure, we have mechanisms for the calcium level in your blood. These mechanisms are either, number one, negative feedback loops, and that's in your notes, number one. Let's look at that, and then we'll look at number two, positive feedback loops. Two different kinds of loops and how they work. What we looked at so far, the most common type of loop is a negative feedback loop. There were negative feedback loops in the examples we just looked at, because we say negative because in that loop, in that circle, the response from your homeostatic mechanism was opposite. It opposes the initial change of the variable. So the stimulus, like it was really cold outside. So what does your feedback loop do? It says shiver, make heat. It's cold, let's make some heat. Let's go opposite of what it was. If you're too hot, then make sweat and evaporate, cool. It's going to go opposite. So we say that's negative, right? Something was happening and it's too much, it's too little. I'm going to go opposite to try to fix it. Opposite, negative. Another reason we say negative is because somewhere in the process, when you have this homeostatic mechanism, there's a point where it has to reduce the effector. I mean, it's all well and good to sweat. It's all well and good to shiver, but eventually your effector that's sweating or shivering, eventually you have to reduce that. You have to stop. You don't want to sweat or shiver and shiver forever, because eventually you're going to get back to normal and you reduce. So let's go back to that first diagram. So right here, once we got back to normal, this is the negative feedback loop right here. Okay. Negative feedback is once we get back to normal, the receptors, okay, remember I was putting those X's on here? Now we have no stimulus here, right? Negative feedback says no stimulus, don't do this, and you can stop sweating, right? Because you don't want to sweat and sweat and sweat, you get too cold. So that's the negative part of it. It's not a bad thing, okay? Negative just means like, okay, number one, I was too hot, so I got to go opposite that and cool off. But then we can also say negative feedback because there's a point where like, okay, sweating was okay, but now I got to stop, negative, stop sweating, right? I tell my students to remember it this way. Negative feedback is not always a bad thing. If you're at work, okay, and you get negative feedback from your boss, what does that say? Your negative feedback from your boss is saying, stop doing that. Don't do that anymore. Why is your boss doing that? It may not feel so good at the time, but he's trying to help you, right? He's trying to help you do the right thing. Stop doing that. And it may be you're horsing around too much, okay? Or, you know, whatever, you're doing too much of something, okay, that wasn't, you know, because I mean, having fun with your coworkers is a good thing, but too much of it is, you know, it's not good, right? Likewise, like think about your studying, you know, like there's a point, you know, if you're spending like 40 hours a week on one class, I might say, you just need a little break. You need to sleep, okay? Too much of that is not good. I might say stop. So and this negative feedback is always when somebody says enough's enough. Okay, it was okay for a horse around and have a good sense of humor, but don't go too much, right? It was okay to sweat, but don't sweat too much. There's a point where, you know, and that's what that negative feedback is. It's not a bad thing. It's just the correction to say like, okay, enough's enough. Think of negative feedback that way. There's a point where a negative feedback is good because that means you're in balance and you don't need to change it anymore, okay? Turn it on, the sweat glands, when you need them, but the negative feedback controls, like so you don't sweat too much or you don't shiver too much. You always need to be able to to kind of come back and say, okay, that was good, but enough's enough. Negative feedback does that. Okay, it's that little correction. Once you get to the right balance, which is what it's all about, it's a little correction. So that's what we talk about. Well, we say, okay, these loops, like I was too hot. I don't want hot. I got to cool down. So that's negative, like going opposite. So you can think of it that way. That's one way to think, okay, I got to do the opposite of whatever was happening. Or the other way to think about negative feedback loops is the fact that, and we're going to talk about this quite a bit, especially in Anatomy 102, that effectors, muscles and glands need to do things, but only to a point. There has to be control. And if you're not controlling it, that's when things get all out of whack. That's when you're the patient in the doctor's office, because you can't control your blood sugar anymore. You can't control your body temperature. Something's out of control. If you don't have control, and we need negative feedback loops to keep us into control, keep us about, because it's not that you're sitting outside in the freezing cold day after day after day. I'm in the house. Okay, the temperature, but I'm too hot all the time, or I'm too cold all the time. Then you go see the doctor, say what's wrong, right? Or I'm drinking the right amount of water, but my blood pressure is still not good. What's wrong, right? It's the control mechanism. Somewhere the negative feedback's not coming in the right way to control it. So it's very important, okay, that you're able to adjust, but then control the effector and have control. The goal is homeostasis, to be in a normal range around the set point. Not exactly this, might get a little high, might get a little low, but you're in a normal range. Okay, might have slight fluctuations, but as it starts to creep up, you correct it. Starts to creep down too much, you correct it, and you come back and you waver around that normal range. Okay, this is like 99% of homeostatic mechanisms do this. Okay, and they have teeter-totters where you get a little up bounce, but okay, we get back. Okay, we're always making a little adjustment. Might get a little bit higher blood sugar, but we bring it back. Might get a little bit lower blood sugar, but we bring it back up. Negative feedback, where you get back in balance most of the time. This is what's happening. There is another kind of feedback loop. This is really rare. Okay, I have somewhere in your notes I put, with little stars, stars next to it said positive feedback loops, not very common. Okay, this is really rare, but sometimes you have a homeostatic mechanism that has positive feedback loop. And we say positive because for two reasons, it either amplifies positive, it's saying more of the change. And in the process, what the effector is doing, it increases. So again, more. Positive means more. Think about if you get positive feedback from your boss, I would be like your boss saying, that was great, do that again. And do that again, that was super. We want you to do that again and again. That's really rare, right? Positive feedback is rare. Usually it's not like that you want a whole lot of something. But sometimes you do that, okay? An amplified response to complete a project. Like you might go, great guns, do a whole bunch of something just to get it done, right? To get it there. It's not real common in the body because you're going to have the effector do more and more and more and more and more. That's rare, but it does happen. Two examples that we're going to see probably in this course. There are other examples, but the two most common are blood clotting and childbirth. Okay, are the most common examples. Now, childbirth, if any of you have children or have been with somebody that's giving birth, okay, the labor process was like more and more and more intense. Okay, because when labor starts, labor and delivery, labor of a child, there is a contraction of the uterus. Okay, it's usually stimulated by, the force of the baby is growing and it's getting bigger and heavier and pushing on the cervix, in that sense, stimuli through neurons. And then it stimulates the brain to release a hormone called oxytocin. And that oxytocin causes the uterus to start contracting because, that baby's just getting too big and heavy. We got to push it out. So the hormone sends a signal to the uterus, and the uterus has smooth muscle that contracts. But once it contracts once, that contraction, that squeezing actually stimulates more pressure on the cervix. And then that sends more signals to the brain, and the brain sends out more oxytocin. You can stimulate labor by Pitocin, an artificial medicine that's like oxytocin. More oxytocin causes more squeezing. So you get an even bigger contraction of the uterus. Well, if it contracts more, that puts more pressure on the cervix. Okay. And you get more pressure, well, you get more oxytocin, and it just builds and builds and builds. The labor, the contractions get more intense and more frequent throughout labor and delivery until the child is born. We completed the task. Okay. So we said, what is this about? An amplified response, more contractions, harder, more frequently, until the baby is expelled from the uterus. The baby is delivered. Now there's no more pressure, right? Because the weight is relieved. And then the whole process can shut down and no more contractions need to happen. Okay. So, but it temporarily, it was increasing more and more and more, the action of the effector. And the change, like we had a little bit of pressure, we actually made more pressure. So you're amplifying, like I had some changing, some pushing on that cervix. Let's make more pressure on the cervix by contracting the uterus. So it amplifies, it's positive, it's more and more, and increases the output. Blood clotting, we'll study this in Anatomy 102, but basically the platelets in your blood, if some of them start to stick together, and you do that sometimes by like, if the blood is moving, you actually put compression on there because you don't want the blood to move. You want that blood to stay really stagnant now, because if the platelets are close together, they might start sticking together. And you, in the clotting, you want them to stick together because they make this like little plug, and it's going to plug up the hole. If a bunch of little tiny platelets get stuck together, that builds a big clump, and that clump can plug up the hole, and that can stop the bleeding. Now, obviously, you don't want your blood to like a whole bunch of it to make a big giant clot and plug up the whole blood vessel, right? But temporarily, we want a little bit of stickiness there, and so that there's a little chunk to like block that opening, right? The break in the blood vessel, and I just plug up the hole. So what happens is some of the platelets start sticking together. If you get some of them start sticking, then they send signals, and then more of them start sticking. The signals they send to each other cause more stickiness. So temporarily, it's like a little bit of stickiness, more stickiness, because, and that's what you do. You put your, compression on it, and you start the clotting process will lead to more blood clotting until the hole is plugged up, and then you don't have leakage of blood anymore. Then it should shut down. Should, right? So, okay. All right. We spent a lot of time on homeostasis. We learned about the feedback loops, both negative, which is most of the situations we're going to look at. We're almost always looking at these teeter-totter, like everything stays in balance. Most of your days of your life, right, everything's pretty normal. But in emergency or unusual circumstances like broken blood vessels, or, you got this large infant child, growing inside of your uterus, right? There's a point where you use a positive feedback loop and have an amplified response just to complete the process. Again, it's homeostasis. You want to get back to normal, deliver that baby, get your blood flow back to normal. It's homeostasis. It's protecting you and getting you back to the normal state. But temporarily, it's positive feedback on itself. All right, a couple more core principles. We're going to go through these a little more quickly. Gradients is a key idea in physiology. And a gradient, and I think I said define gradient. Well, here's some examples. Like here's a heater, and it's really hot, 80 degrees by the heater. If you move away a little bit, it's only 75 degrees. If you move away a little more, it's only 70 degrees. There's more heat by the heater and less heat away from the heater. So more of something, less of something. Here's another example. A pill, it has a chemical, a medicine in it, concentrated, lots of medicine right here. But as it dissolves, there's some medicine that's moving away from the pill as it's dissolving. And so right in here, you've got some medicine. If you look further out in the beaker, out here, there's just a little bit of medicine. So again, more in one area, less in another area. Or the last example they're showing here is air pressure. Before this plunger was, pushed, like you may have had the same pressure inside and outside, but as you depress the plunger, you're concentrating the air in here, and that air is under more and more pressure because smaller volume, pressure gets higher. So now you might have higher pressure out here and lower pressure out there. Okay, that's a gradient. A gradient is when, okay, and it can be heat, it can be chemicals, it can be air, it can be liquid pressure, like blood pressure. You get more blood inside your blood vessels. That's a gradient, right? More blood in your blood vessels, maybe less fluids outside the blood vessels, right? And a gradient, more of something in one area and less in another area. Okay, now a key point about this is that the areas have a connection such that some of the stuff, whatever you're talking about, whether it's the medicine or the air or the blood or the heat, right? Some of the stuff can move, okay, from the area of high concentration to the area of low concentration. Okay. That's a key point about gradients. So substances move from high concentration to low concentration. What, you know, that's why you have a heater in your house, right? There's a whole bunch of heat there, and you're hoping that heat's going to move out and spread out throughout your house and heat up the whole house. And it does, right? Heat moves. Heat just does that. It radiates. You put the pill in water and it dissolves and it spreads out. It's called diffusion. We're going to talk about that this semester. But the molecules spread out. Here, this is closed, but if we take the cap off this little plunger, what's going to happen in the air? It's going to push out really hard and fast, right? If you took the little cap off here, because air does that. It doesn't like to be concentrated. When there's a pressure, it's going to try to move from high concentration to low concentration of pressure. This is really important for stuff to happen in our body. Physiological process. How does the air move into my lungs? How does the air move out of my lungs? Okay. The heat that's in my muscles. How does it get from my muscles to my brain, to my skin? How does the sugar in the food that I eat get spread out, like into my blood and into my body fluids? You know, how do things move around? A lot of it is gradients. So this is a key principle that drives many of the processes. We'll see gradients in action. So key point. So it's a gradient. A gradient is having more of something in one area and less in another. You know what I think of sometimes? I think of gradients. Ombre. Okay, look at the shirt. It's got a whole bunch of color down here and less of the red up here. And it kind of fades in between. That's classic gradient of color. So it can be the dye in her shirt. It can be temperature or heat. It can be pressure of air. It can be pressure of fluid. It can be, you know, chemical, sugar in your blood. So, but you have more of something in one place and less in another, and that's a gradient. And what's going to happen? It's going to try to even out. And that evening out is good because that's what causes some of the reactions and the activities in our body. All right. I think we're on last topic here. Core principle, last core principle, cells must communicate. We said there are trillions of cells in your body. One cell has to be able to communicate to another cell. For example, we said in homeostatic mechanisms, the neurons, the hypothalamus send a message to, let's say, the smooth muscle cells. Okay, how do they, you know, the smooth muscle cells around the blood vessels? We said that's what opens up the blood vessels and allows all that blood to flow in my skin and make me red and release the heat from my body. How How did those neurons send a message to the smooth muscle cells? They have to communicate. The neuron cell and the muscle cell have to communicate. How are they going to communicate? Usually electrical signals or chemical signals. We're going to study both. This semester, we're going to learn about action potentials are called. Action potentials happen inside the neuron, like so from the dendrites down through the axon, electrical signals are being sent. We looked at the ends of these neurons and we saw like one neuron and sending little yellow dots, we said little chemicals called neurotransmitters going from one cell to another. Here's another one with a neuron sending a chemical neurotransmitter signal to a muscle cell. So chemicals. And how do those chemicals move? Usually when you have a lot of chemical and not so much chemical, the chemicals move according to the gradient, right? That's how that works. Okay, gradients drive the movement of the neurotransmitters. Hormones are another one. It's a chemical like from one place in your body, and then the chemical goes through your blood and spreads out. I have a whole bunch of it here. Well, it spreads out through your blood and goes to other places and sends a chemical message, causing a change in another part of your body. So electrical signals, chemical signals are usually the way our cells can talk to each other, okay? The way they can send messages back and forth. So we'll be looking at those key ideas, like how neurotransmitters work, how hormones work, and other chemical signaling and electrical signaling in the course of this anatomy and physiology studies. Okay, and at the end, she has a little table. And she has little symbols, like the feedback loops, think of the circles, you know, this receptor stimulates the control center, stimulates the effector. The information is fed back, negative feedback or positive feedback, back to the receptor to regulate. Or structure and function, she has little gears. Gradients, we see lots of dots and a few dots, right? Gradient. Or here's our cell, like sending those chemical signals from one cell to another. Watch as you read your textbook and you'll see these little symbols pop again. So, hey, what am I talking about here? This is gradient. Remember what a gradient is? This is how this works. This is how, for example, your muscle cells can stimulate a change for contraction, okay? Or in the muscle cells, one protein hooks to another protein, how they hook together, the shapes of their little parts of the proteins, how they function. So she'll use these symbols to kind of cue you in again on these key ideas that she taught in the first lesson, in the first chapter, and we'll see these main themes again and again as we study how the body works, physiology. Okay, so that wraps up your notes for chapter 1. Refer to your textbook, add additional notes as you're reading, as you work on dynamic study modules. Keep adding to, hopefully you have like a notes packet that you can add more to, or in your text, in your notebook as you're writing things down. Keep adding more notes. We've got a lot from this lecture, but as you do more work in your reading and in your studying and doing lessons and activities, add to your notes so that you'll have a really good set of notes to study from. And then next time, go to, again, the modules. Be ready to move on to chapter 2. And you'll look for the lecture notes for chapter 2, print those out, save them on your computer, and get ready to read along and follow along. You can go to the lecture recording for chapter 2 and start working on notes for chapter 2. All right, thanks for listening. I will see you soon.